I will show how you can apply microbes to ferment the main sugars from lignocellulosic substrates. As explained before, lignocellulosic substrates contain mainly glucose and xylose. Most microbes can handle glucose, but many have issues with xylose. Baker's yeast, Saccharomyces cerevisiae, is not able to ferment C5 sugars under anaerobic conditions. It can only convert xylose via xylitol to xylulose. As the first step is consuming NADPH and the second step is producing NADH, there is a redox imbalance. Under aerobic conditions, NADPH and NAD+ can be regenerated. Under anaerobic conditions, NADPH and NAD+ cannot be regenerated, and as a result, yeast cannot grow. Bacteria convert xylose to xylulose phosphate in two steps involving xylose isomerase and xylulokinase. This would also be the preferred pathway for yeast. The only enzyme missing in yeast is the xylose isomerase. However, attempts to introduce bacterial xylose isomerases were not very fruitful. Discovery of a Pyromyces fungal xylose isomerase was the turnaround point. This enzyme turned out to be functional in yeast. This was a great breakthrough in the development of ethanol production from lignocellulosic substrates. When using bacteria to convert xylose, there are other issues that may interfere with the optimal use of lignocellulosic feedstocks. E. coli has the capacity to use xylose. However, a process called catabolite repression shuts down the use of xylose when glucose is present. Therefore, it will first consume glucose and then start to consume xylose. As a consequence, E. coli needs to be engineered to allow simultaneous consumption of glucose and xylose. A third way to metabolize xylose can be found in some lactic acid bacteria. They use xylose via the phosphoketolase pathway, generating equimolar amounts of lactate and acetate. This is not very useful if you would like to use them for lactic acid production from lignocellulosic substrates. Fortunately, there are bacteria, such as thermophilic bacilli, that use the pentose phosphate pathway to convert C5 sugars to lactate. This allows for a complete conversion where 3 C5 sugars are converted into 5 lactate molecules. These thermophilic bacilli, such as Bacillus smithii, Geobacillus and Parageobacillus species, are particularly suited for lactate production, but also for production of ethanol or other products. They can be isolated from compost and are specialized in fermenting lignocellulosic sugars. In contrast to E. coli, they do convert glucose and xylose simultaneously. So now you have learned that different organisms have different issues that need to be solved to allow for simultaneous use of glucose and xylose sugars. Yeast needs a different xylose conversion pathway. E. coli has catabolite repression that prevents the simultaneous use of xylose and glucose. The phosphoketolase pathway of lactic acid bacteria is limited in its use, as it results in a mixture of lactate and acetate. Thermophilic organisms are very well suited for simultaneous saccharification and fermentation of lignocellulosic substrates. For all microbes, further strain development is required to have a more economic process and to facilitate the transition to the bio-based economy.